So we're going to take look at our biological molecules. We're going to look at 4 different biological molecule groups. The first group that we'll take a look at are the carbohydrates. So the carbohydrates, typically we think of these as being sugars. The technical term here, though, is saccharide, or saccharides, for the different sugars. Then we'll take a look at our lipids. We also call these fats. And so our lipids are actually fats. And we'll take a look at the different types of fats or lipids. Then we'll take a closer look at proteins. And then we'll take a look at our nucleic acids. So nucleic acids meaning our DNA and RNA. All right, so let's go ahead and jump into our carbohydrates. So actually first, before we talk about carbohydrates, each of these biological molecules have a similar type of structure in that we need to talk about what a monomer is and a polymer. So a monomer is like a building block. So the monomer is a building block, meaning that it's a single piece of that molecule, and a mono meaning single. So monomer is a single building block piece. And then polymer, poly meaning many. So what we're going to do is we take several of these building block pieces and we put many of them together to make our polymer. And so as we're looking at these different biological molecule groups, the carbohydrates, there are different monomers of carbohydrates. So there are different polymers of carbohydrates. For lipids, there are different monomers for lipids and different polymers for lipids, and for proteins and for nucleic acids. So it's important that you understand what the monomers are of each of the biological molecules as well as the polymers. So if we're getting now into our carbohydrates, we look at our carbohydrates. Our monomer for the carbohydrate, our basic unit is called a monosaccharide. So monosaccharide. Remember, mono means single or one, and so mono for singular and then saccharide for sugar. So our monosaccharide is a single sugar. And the one that we talk about the most is glucose. So a single glucose molecule would be a monosaccharide, single sugar. There are other monosaccharides that you should be aware of. Some other monosaccharides are fructose. Also, we have galactose. And then our last one is actually ribose. So these are our 4 different monosaccharides. So you should know the different monosaccharides. So this is our basic unit of a carbohydrate. So it contains carbon, hydrogen, and oxygen. So if we take a look at these 3 different elements, carbon, hydrogen, and oxygen, kind of shorthand name for carbohydrates is the CHO. If you think about those 3 different elements, can you think about whether or not this molecule is going to be polar or if it's going to be nonpolar? Right, so if we're taking a look at something like oxygen, which is pretty electronegative, especially compared to hydrogen, which we've talked about as far as water is concerned, and remember the carbon and the hydrogen are similar in electronegativity, what we see here then is a polar molecule. So our carbohydrates are going to be polar molecules. And what did polar mean? Remember, that meant that it interacted and mixed well with water. So then we had the H and the O from the water, H's and the O from the water, that are going to interact well with our polar molecules, including our carbohydrates. So our carbohydrates, our sugars, can dissolve in water. And we actually know that because we know sucrose, which is our table sugar, does dissolve well in water. Water makes it a polar molecule. So our glucose is our basic unit here. It's the most important energy source in cells. Our galactose is an isomer of glucose. Isomer meaning that it has the same pieces, they're just arranged differently. We find galactose, if you can see this term lactose, perhaps that's familiar to you. Actually, I can write this down here. So glucose. Glucose is our most important energy source. Most important energy source. Galactose. The giveaway here is the lactose, perhaps you've heard of that, is our milk sugar. So we find this in our dairy products. And in some legumes. Then our fructose. Fructose is another isomer of glucose, meaning it has the same pieces, the carbons, the hydrogens, the oxygens, just arranged differently. And fructose is the kind of sugar that we find in fruit. So fructose is in fruit, as well as honey. So it's our sweet sugar. And then our last one, ribose. Ribose is the monosaccharide that we find in our DNA and RNA. And we'll talk about that more once we get to our nucleic acids. So we have our monomer. Remember, our monomer is our single piece, our single unit of our carbohydrates. In this case, we're talking about our monosaccharide. If we then take two of our monosaccharides and put them together, what we get is called a disaccharide. And actually, really quick, I think I'm going to draw just a quick image here of glucose so we can kind of get an idea of what glucose looks like. So it's a hexagonal shape here when it's in its ring structure, ring form. We have some hydroxyl groups here. Hanging off the edges again because it's a very polar molecule. You can imagine here this would be our partially positive end, our partially negative end here hanging off the edge of our glucose. This guy here, I should probably have just left a space right there. That is actually an oxygen in there. And then off of here we've got another little chain with some carbons, some hydrogens, and another OH group. So this is our basic structure right here of a glucose molecule. And so then when I'm drawing and we're taking a look at our polymers of carbohydrates, our polysaccharides, I'm probably going to shorthand it, kind of like a circle, semi-hexagonal shape. But what I'm implying here is this structure that I've just drawn. So this is our glucose structure. So getting back to where we were, our monosaccharides are single sugars. These are four single sugars, the glucose, galactose, fructose, and ribose. When we take two of those single sugars and we put them together, we get a disaccharide. Di meaning two, and then saccharide again meaning sugar. So our two sugars. So we take two monosaccharides and put them together. This is a reaction between the two. And then when we join them together, we remove water. And we have our different disaccharides. So the first disaccharide you should be aware of is lactose. And lactose is a combination of glucose, and can you guess the other one? Galactose, yes. So we take our glucose molecule, we have a bond between the glucose and the galactose, and we call that lactose, which is a disaccharide. The other disaccharide that you should be aware of is sucrose. And sucrose, we already mentioned, is table sugar, and that is a combination of glucose and the other one, right, fructose. So we have lactose, which is our milk sugar. So this is our milk sugar. We find it's a combination of glucose and galactose, and sucrose, which is our table sugar, and that's a combination of glucose and fructose. Now, if we go further and we add more monosaccharides to this, we don't just have two getting us to the disaccharide level. If we have many saccharides all in a chain, so I'm going to draw that. So this is kind of, if you can assume this is our glucose molecule, then we have another glucose molecule. This right here would be our disaccharide. Now, if I added more, then we no longer have a monosaccharide, which would be a single saccharide. We no longer have a disaccharide because it's more than two. What we have here is a polysaccharide. Poly meaning many. So this would be an example of a polysaccharide. And we have a couple of polysaccharides that you should be familiar with. So the first polysaccharide you should be familiar with is called glycogen. Glycogen is our main form of stored glucose. In animal cells. So our polysaccharide, many saccharides, lots of saccharides all together, our main form in our bodies, in animal cells, is called glycogen. So when we are eating something, think of something that is high in carbohydrates. So you can think of something like bread, which has a lot of carbohydrates. You can think of something like a fruit that has a lot of carbohydrates. You can think of something like a candy or soda, although that's kind of high fructose corn syrup. But lots of different things we can think of when we think of things that have high amount of carbohydrates. When you eat that, our body is going to break that down. It's going to break it down into individual glucose molecules. So we're going to eat it in a polysaccharide form, a long chain form, storage molecule in those foods, which we'll talk about in a moment. Our body is going to break that down. We're going to break it down into individual glucose molecules, and we're going to absorb those glucose molecules into our cells. If our body needs energy right now, if we're tired and we need an energy replacement, we move that energy, those glucose molecules, around in our body so they can be used for energy. If, however, we eat more glucose than we actually need at this very moment for energy, then what happens is we store it for later. And our liver does this for us. Our liver stores all of these extra glucose molecules that we don't need immediately, and it stores it in the form of glycogen. So we take our glucose that we've broken down in our intestines, we absorb it, it goes throughout the body in our bloodstream. Once it gets to all of the different organs, all the different parts of our body, and our body says we're all filled up, we've got enough glucose, we've got the energy we need, then it's going to go to the liver. The liver is then going to take the individual glucose molecules, stick them together, and then form what we call glycogen. And then it's stored in the liver as glycogen so that later, hours after you've eaten your meal, if now you've used up the energy stores inside the individual cells, the liver can then break down that glycogen, breaking it off piece by piece and sending those individual glucose molecules out into the bloodstream. So glycogen is our storage form in animal cells of glucose. However, when we talk about what we first ate, right, we ate a bread roll or we ate a potato or something high in carbohydrates. When we ate that, we didn't eat it as glycogen. I said that we broke it down into individual glucose molecules, which we did, but the way that we ate that in these examples is via starch. So starch is actually the main form of stored glucose in plant cells. So the main form of stored glucose in plant cells. And while that's not important for storage in our cells because we don't use starch as our storage form, it's important because that's what we're eating. We're eating starch because we're eating that from plants. So we're eating starch, which will look similar to this molecule up here because we're just talking about individual glucose molecules attached to each other. We eat it as starch, our body breaks off these individual glucose molecules, and then we store the glucose as glycogen and break it down for use later. So our two polysaccharides so far that we have are glycogen, our main storage form of energy, and starch, plants' storage form of energy. Third one is cellulose. Now, cellulose, again, is a polysaccharide, which means that we have several glucose molecules that are all attached to each other. But cellulose is not a storage form of energy. Cellulose is not digestible by humans, and what it is a long chain of glucose that is used for form. This is actually what we call fiber. So the common term of fiber, one type of fiber, is cellulose. So cellulose is actually the plant structure molecule. So plants, they take glucose, they make glucose, and what they do with it is, one, they store it in their own form as starch so that they can use that energy throughout the day as they're using up energy and they need more glucose. But they also take glucose and they put it in the form of cellulose, and cellulose is what's going to give structure to each plant cell. And that's important for us in human biology because, of course, as we're eating plants, we're eating not only starch, which gives us glucose as a stored form, but we're also eating cellulose. And cellulose is actually our non-digestible fiber form of carbohydrate. And then there's one last polysaccharide you should be aware of, and this is called chitin. And then chitin is also a structural carbohydrate. Now, this structural carbohydrate actually is found mostly in the external layer of insects. So this is the exoskeleton of insects. Wow. Let's start over with that here. So, exoskeleton of insects. There we go. Exoskeleton of insects. We also find chitin in things like mushrooms. So we do find it in things like mushrooms, also lichens, not that we're eating too many lichens. So we do get some of this in our body, but again, it's not something that we can digest. It's not something that we're able to use for energy. So these are 4 polysaccharides that you should be aware of. So again, our monomer of carbohydrates is the glucose molecule or other monosaccharides like fructose, galactose, and ribose. Then we have our disaccharides. Our disaccharides were lactose and sucrose. And then we have our polysaccharides. And our polysaccharides were glycogen, starch, cellulose, and chitin. Then we are going to move on to our lipids. So lipids, also known as fats. We have different forms of lipids. So the main function of our lipids is going to be energy. Similar in that way to carbohydrates. So our carbohydrates, the main function of carbohydrates is energy. Our main form of energy in our bodies is glucose. Now, carbohydrates and glucose are our quick form of energy. So as we eat food, the easiest thing for our bodies to use for energy is glucose. It's our main form. So our body's able to easily move glucose throughout the body and use that for energy. We're able to easily store it as glycogen and then retrieve it again, break it apart, and get the glucose from that. And what we do is we use that as our first source of energy. However, once we get to a point where we've used up the free-floating glucose in our body, and we've used up the glycogen that our liver has stored from our last meal, if we're many, many hours out, say you've gone to bed, you've woken up the next morning and you're hungry, at this point, we've used up all of the glucose storage. We've used up all of our glucose that was floating around and our glycogen. And so your body then is going to move into using lipids or fats for energy. So this is our long-term energy storage. Our carbohydrates are short-term, and our lipids are our long-term. And so our monomer, again, we have our monomer and polymer structure. Our monomer is the fatty acid, which we'll take a look at in just a moment. Our fatty acid, or depending on the type of lipid we're looking at, it'll be a sterol ring. And that'll be more clear in just a moment. When we're talking about lipids, what we're talking about mainly here is carbon and hydrogen. So not as much of the oxygen here. There are only a few oxygens, and those are in very select areas. So if we're looking at mainly our fats being composed of carbon and hydrogen, do you think that this molecule is going to be polar or nonpolar? Right, nonpolar. Right? Because remember, our electronegativity of our carbon and our hydrogen is pretty similar. So we're looking at a nonpolar molecule. So our lipids are nonpolar, which means that they don't dissolve well in water. And we know that if we pour some sort of fat, like an oil, on top of water, they don't mix very well. Same thing with butter or with lard, which are saturated fats. So we have our three categories of lipids that we're going to take a look at. So we'll take a look at our triglycerides, our phospholipids, and our sterols. So when we talk about our fatty acids, we'll go back to our monomer for a moment, and then we'll look at our monomer and how it relates to each of these different types of lipids, the three types of lipids. So our monomer, again, is called the fatty acid. We actually have different types of fatty acids. We have our saturated fatty acids, and we have unsaturated fatty acids. When we look at a fatty acid, really what we're looking at is a whole bunch of carbons. So it's a carbon chain. And our carbon chain has a bunch of hydrogens attached to it. Let me get my hydrogens in here since I drew so many carbons. So we have a carbon-hydrogen chain is what we're looking at for our fatty acid. So our fatty acid chain then here is this here. So we have a chain of carbons all with lots of hydrogens on them. So the difference between our saturated and our unsaturated fatty acids is actually the amount of hydrogens that we have on our chain of carbons. When we have a chain of carbons that is completely saturated with hydrogens, that's our saturated fatty acid. Saturated meaning, right, completely filled with. We have as many hydrogens as we possibly can on all of the carbons. If we recall, carbon can make 4 bonds. So if we see each carbon here, we have a bond between this carbon and this carbon and the next carbon. But then we have our hydrogens filling up the last 2 bonds. So if we look at, let's say, this carbon here, right, we're bonded to the carbon behind it, we're bonded to the carbon in front of it, and then that only leaves 2 more bonds. Both bonds are going to be 2 hydrogens, and we see that same thing with every carbon here. So if we look at this carbon right here in the middle, we have a bond to the carbon, to the left of it here, carbon to the right of it, and then that leaves 2 bonds that we can make with hydrogens. And both of those bonds are bonded to 2 different hydrogens. So this right here is actually a saturated fatty acid, where we are completely filled with hydrogens. When we have a saturated fatty acid, it looks just like this, in that it is very straight. Because we have single bonds between the carbons, that means that we have a very long, straight chain. And then we have the hydrogens hanging off of the ends there. Now, if we take a look at what an unsaturated fatty acid looks like, that means that we are not saturated, right? Unsaturated means we are not saturated with fatty acids or with hydrogens. So an unsaturated fatty acid means that there are 1 or more double bonds. So then what we see is we have our carbon chain. So we have some carbons here, but in here we end up having and I'll put 1 in for now, a double bond. So this is our double bond between the carbons. And so over here we have our maximum number of hydrogens, right? So if we look at this carbon has a bond to a carbon behind it, in front of it. It has 2 bonds left. Both of those are to hydrogens. Same thing with this one. And same thing with these over here. So we still have our carbon-hydrogen chain. However, we have this double bond here. So this double bond now is taking up 2 bonds between this carbon. So let's see if we look at this carbon that I'm highlighting here. It has 1 bond to the carbon behind it and 2 bonds to the carbon in front of it. How many bonds does that mean it has left? Right, it only has 1 left because we have 1, 2, 3, and then 4. So we only have 1 that it can be attached to that one. And then we see the same thing with this carbon here, right? So it has 1 bond to the carbon in front of it, 2 behind it, which means that it only has 1 space left for a hydrogen. So we are missing some hydrogens here because we have this double bond, which means we have an unsaturated fatty acid. It's not completely filled with hydrogens, it has a double bond. And then what we could also see, because we said 1 or more double bonds, is we could have another spot, let's say right in here, that has a double bond as well. Right, so we could have 1 or more, but then of course these guys wouldn't have these hydrogens because they're not saturated. And then it would be attached to another carbon, and let's say this carbon has its hydrogens. Also notice that when I drew the saturated fatty acid, it was a nice straight chain here of carbons with lots of hydrogens attached to it. When I drew the initial, with the exception of this one, the initial chain here of our unsaturated fatty acid, that it is not straight. And that's because of this double bond. The double bond actually pulls on the carbons in such a way to where it adds a kink to the chain. And so this is what makes it an unsaturated fatty acid, is the double bond being in there and then this bend or this kink. This is important because a saturated fatty acid, we can have them lined up right next to each other, very, very close to each other. And we know that this is true because if we look at something like butter compared to oil, for example, butter is a saturated fatty acid, and it is solid at room temperature. Versus oil, if we look at the oil chains, we're going to have a lot of space in between them. Right, because if we have these chains, they can't go right next to each other like we saw with the butter. There's kinks in there, so they can't sit really, really close to each other. What that means is that this ends up being more fluid. Since we can't have everything sit very, very close to each other, it's not more stable, it's not so solid. And so then we have something like an oil here versus butter up here. So a good way to tell whether or not something is a saturated fat versus an unsaturated fat is just at room temperature, is it liquidy or is it a solid? So if it's a solid, then it's likely a saturated fat. If it's more liquidy, because of these kinks, it allows space between the saturated or between the fatty acids, then it's likely an oil or an unsaturated fatty acid. So we have those 2 different types of fatty acids. The fatty acids then are important as we move on to our first type of lipid. Remember, our first type of lipid was the triglyceride. Our triglyceride is our long-term energy storage. So we already mentioned lipids are used for energy storage, but long-term. So what we do in our body is we take these fatty acids, which were our monomers, and we build them to make our polymer. Our polymer is the triglyceride. We take our fatty acids and we stick them to what's called a glycerol backbone. So we have our glycerol backbone, which is actually just a series of 3 carbons. I'm going to leave off some of the other pieces that would be hanging off here just for simplicity. There's some carbons and some hydroxyls here. So we have our glycerol backbone, and our glycerol backbone then is attached to our fatty acids. So attached over here, we end up having our fatty acids. So we can say that, let's say we have an unsaturated fatty acid, that's why we have the kinks. We can have a saturated fatty acid, and then we can have another unsaturated fatty acid, for example. So a triglyceride can contain both saturated fatty acids and unsaturated fatty acids, depending on what's floating around in our body. But the triglyceride, tri meaning 3, and glyceride meaning glycerol backbone, is our completed structure, our polymer. So tri meaning 3, because we can fit 3 fatty acids in here. And then we have our glycerol backbone. So that's our triglyceride. So we take our 3 fatty acids, we attach them to a glycerol, and then we're able to store those in our fat cells. So that's the structure of our first lipid, is taking those monomers, the fatty acids, and attaching them to the glycerol for our long-term energy storage. The second lipid that we're going to talk about, remember, is called the phospholipid. So the phospholipid, what it's most important for our purposes, and in all purposes really, for not only human cells, but also other animal cells, but also plant cells, bacterial cells, the phospholipid is to form membranes. To form membranes. Our phospholipid structure is that we have a glycerol, which we just noticed in our triglyceride. We have a glycerol, but it has 2 fatty acids attached to it. So instead of tri, meaning 3 in our triglyceride, now we have 2 fatty acids attached to a glycerol. Our glycerol is hydrophilic. Remember what hydrophilic meant, philic meaning loving. So our glycerol is hydrophilic, which means that it mixes well with water. Our fatty acids are hydrophobic. So remember, this means that it doesn't mix well, and we've been talking about that this whole time with our lipids already. So remember that our phospholipid then is called amphipathic. And it's amphipathic because it has both a hydrophilic region and a hydrophobic region. The way this is generally drawn is this up here. This is our head, the glycerol head. And then we have our fatty acids. And this is the structure of our phospholipid. And this is an important molecule because it is what is going to form all of the membranes on every one of our cells. So all of the cells in our body have a membrane, a plasma membrane on the outside that we'll take a closer look at, and that is largely composed of phospholipids. What we see then is that our hydrophobic region, remember our water disliking region, is going to try to get away from the water, try to get away from any type of water region, which of course our bodies are composed mainly of water, so there's a lot of it, and they try to get away from the water and kind of quarantine themselves. And then our glycerol head is going to interact with water. So what we see is that we generally form what's called a bilayer of our phospholipids. So we have our heads like this, and then our fatty acids. And then we see on the opposite side, bilayer meaning 2 layers, bi meaning 2. So we have our hydrophilic heads all lining up next to each other, and our hydrophobic tails are quarantining themselves in this hydrophobic layer here in the middle. Out here we have our water, and out here we have our water. So this would be the water, say, inside the cell. And this would be our water outside the cell. And then this would keep going and form our plasma membrane around our whole cell. So we see that we have this part that interacts with the water and then this part that does not interact with the water. And we're going to go into more detail when we talk about our cells specifically. So at this point, it's just important that you understand that we have this glycerol head with 2 fatty acid tails attached to it. Our glycerol head is our hydrophilic region, water-loving, and our fatty acids, again, are hydrophobic, or water-disliking. Our third lipid that we're going to take a look at are the sterols. The main sterol that's important for us is cholesterol. So a sterol is used for structure. Mainly the cholesterol is used for structure in our cell membranes and for communication. Communication because some of our hormones are sterol hormones. So things like progesterone, estrogen, and testosterone. Also some other ones like cortisone. So these are our sterols. And the structure of a sterol, this is where we get back to the monomer. The monomer of a sterol is actually the sterol ring. So I'm going to just kind of draw a quick structure here. So this is a carbon chain. Remember the glucose piece. We can imagine each one of these corners is a carbon. And then they have hydrogens kind of sticking off of them. So I'm going to actually remove the hydrogen so that we don't get confused. But I just wanted to be clear that these are our carbon rings, similar to what we saw with our glucose, but different because we don't have our hydroxyl groups hanging off of the ends here. So this right here is cholesterol. So you can see that we have multiple rings here. These are made up of carbons and hydrogens. And these are all hydrogens hanging off of every single one of these carbons. This right here is the only piece that has something that's not a carbon or a hydrogen. So you can see, as we mentioned, lipids are hydrophobic. This whole area here is hydrophobic. It's all carbons and hydrogens, and it's made up of our sterol rings. So our cholesterol is important because it keeps our plasma membrane fluid. It actually acts as a kind of like our unsaturated fatty acids, how they are kinked, and so they have space between them, and that allows them to stay fluid. Our sterols, or specifically cholesterol, you can see that this is kind of kinked in that same way, and so when we put it in with all of those phospholipids, it's going to maintain the fluidity of, let's see, we're going to pretend our cholesterol here. It maintains the fluidity of the cell because these guys are not able to be packed so close together. So it allows it to keep our plasma membrane fluid. So we have cholesterol as one of our sterols. And then, as I mentioned, these guys up here, we have our steroid hormones. And that's important as we get to the endocrine system and talk about the different hormones. We'll be talking about the different types of steroid hormones. That's all I'll say about that right now. The last type of sterol, so we have cholesterol, we have our steroid hormones, and then we have some lipid vitamins. And our lipid vitamins are A, D, E, and K. So again, we will be talking about these vitamins as well, but not in as much detail right now. So that is the end of our lipid section and our carbohydrate section. And I'm going to post another video with our proteins and nucleic acids so we can be not so long on this video here.